As we have seen in our previous module that during the isolation process we mostly get a mixed culture. We have to need for the pure culture. So in order to get the pure cultures, the isolation method mostly commonly used to get the pure culture. That is the streak plate method. In next slide you will see that how that is streak plate method. So a sterile inoculation loop is dipped into the mixed culture that contain more than one type of the microbes and is streaked in a pattern over the surface. There are different patterns you can streak over a plate on the nutrient medium. So as the pattern is traced, bacteria are rubbed off and the loop on to the medium. So the last cell to be the rubbed off the loop are far enough apart to grow into the isolated colonies. So these colonies can be picked up with an inoculating loop and transferred to a test tube of the nutrient medium to form a pure culture containing only one type of bacterium, which we called as a pure culture. So you can see in this plate that how we have the streak plate method for a pure culturing. So you can see that here first we dip and that we have a different line, and then this is the second zone, and then it go to the third zone, and then is the fourth zone. You can see here that the different colonies we get that have isolated colonies. Otherwise you can have the different lines in which so many colonies are overlapped to each other. Here another pattern of the streak plate. You can see that here is you can just streak here, and then you can go by this, and then this, and then you can have that. On this range, in this zone, we have a very isolated colonies. By the same case, we have a very selected medium, and then we streak, and then we have a different isolated colonies. So we can pick these colonies as a pure culture. So these are all the overlapped colonies. So for the having the isolated colony, we assure that these are the pure cultures or the pure stains. So the most desirable isolation medium would be one which select for the desired type and also allow a maximum genetic expression. So the culture grown on such medium could then be used directly for the screening. So however, it is more common that once isolated the organism are grown on a range of the medium designed to enhance the productivity. Because if we have a different microorganism, here I want to clear that if we want to isolate, there is the first in experiment we have a number of organisms. So suppose if we talk about the fungal organism, that might be have Aspergillus niger, Trichoderma, etc. So if we have a different isolates having belonging to the same genera and a same species like Aspergillus niger from different habitats, so we can call them the stains. So if we have Aspergillus niger from different habitats, that can have the different potential of the product formation according to their acclimatization of that environment. So we have to select that we have to optimize then by changing different media in which that organism can produce a maximum product which we need through fermentation process.